Hello, I'm Richard Young, your host with Facundo Batista and Lena Fayan for MIT's course on COVID-19, SARS-CoV-2, and the pandemic. The purpose of this course is to learn about the virus and the pandemic from the world's top scientists, but also to learn what the world's scientists are doing about it. Today, I have the pleasure of introducing you to one of the world's scientific leaders, Dr. Eric Lander, who, with some of his colleagues, is going to tell us what they've been doing to help us safely navigate this pandemic. Eric is a consummate scientist, a 1987 MacArthur Fellow, widely regarded as the Genius Award. Eric's a mathematician, a geneticist, a leader of the Human Genome Project, a creator of a remarkable research institution, an entrepreneur, and an advisor to presidents and popes. He's a professor of biology at MIT, a professor of systems biology at Harvard Medical School. He's a former member of Whitehead Institute, and he's the founding director of the Broad Institute of MIT and Harvard. He was the co-chair of US President Barack Obama's Council of Advisors on Science and Technology, and he advises Pope Francis. Eric, thank you and your colleagues for joining us.
Oh, Rick, thank you. Thank you so much for that wholly unnecessary introduction. It's a pleasure to be here. And I just want to say how grateful I am to you and Facundo and everybody for organizing this course. I have been an avid student in it and enjoying it very much. And it's a real treat to be able to provide a last session of the course talking about work going on, here right at MIT and Harvard as part of what are many, many responses in this community to the pandemic. So the goal today is I'm going to turn to two colleagues who I'll introduce in a second to describe a project that arose in March when one of the faculty members at the Broad, Dr. Deb Hung, who is also a physician at the Brigham and Women's Hospital, called up the head of the genomics platform at the Broad, Stacy Gabriel. Deb had just come out of the meeting for the COVID-19 team at the Brigham. Stacy was actually driving to yoga class at the time, as she has publicly stated. And Deb said to Stacy, we have to stand up viral testing. And Deb and Stacy conspired over the coming days, together with the two people I'm about to introduce, to stand up a testing operation, which has grown and grown and grown. It was not anything that we had been involved in of infectious disease diagnostics. But as with so many people around the world, we're all involved in things that we never expected to be involved in. And so maybe the point of this last session is just to give one story about how groups can step up and do things in extreme situations, and maybe to learn some things about the planning, the processes, the challenges, et cetera, of doing it. So I am in awe of the two people I'm going to introduce, my colleagues. We'll all kind of do this and be available for discussion. But Sheila Dodge is the general manager of Broad Genomics, and she is an institute scientist at the Broad, which is a very high title for the best scientists who are also deeply involved across the Broad. She's been at the Broad for more years than the Broad has existed, namely for more than 20 years when she was working on the Human Genome Project. But she's also unusual because in addition to her scientific background, she has an MBA from the Sloan School, which she earned during the time over the last 2 decades that she has been doing this. So she brings the dual perspectives of a molecular biologist and someone who understands really, productions and operations and things like that. And you'll probably see how that has managed to be really important. And she's going to be joined by Niall Lennon, who's the Senior Director of Translational Genomics and Product Development at the Broad. He too is part of the genomics of Broad Genomics, and he too is an institute scientist, this highest designation of people. Niall hasn't been around for 20 years, just like 14 or 15 years or so. So he's a newbie. No, I'm joking. Niall is a tremendous pillar of this whole operation. He joined in 2006 and played a central role in scaling up massively parallel sequencing. He has his PhD from University College Dublin, did postdoctoral work at the Harvard Medical School and the MGH, and he has an executive certificate in management from the MIT Sloan School as well. And he's an adjunct professor of biomedical engineering at Tufts University. So I am blessed with amazing colleagues, and all credit that goes for what's been done goes to them and to Stacy Gabriel, the director of the platform. And I'm going to turn over to Sheila, I think, who is going to drive.
Great, thank you, Eric. And so what we'd like to do today is give you a little bit of a lab tour, a little bit of the journey that we've taken over the last 6 to 8 months in scaling the testing capabilities that we have at Broad. So Niall and I will sort of tag team, and Eric will jump in as needed as we go through this. But to give you sort of a snapshot of where we are today, we've completed over 6.5 million COVID tests since March at the Broad Institute, and we'll talk through kind of how we were able to achieve this scale in actually a very short period of time. And you can see that scale up really started in the last 4 months or so. So that's where most of the testing has happened, was since the summer. But the early days of testing really taught us so much about how to do this and how to think about scale in this type of viral testing, even beyond the doors and the rooms of Broad. So our current capacity today is over 100,000 tests per day, and we have achieved that a small number of times so far. We actually reserve some of our capacity for surges and sort of unexpected needs. So you'll see us often running closer to 80 or 85,000 samples a day. We are now serving over 900 collection sites. So this ranges across a variety of different participants, including state Stop the Spread sites, as well as nursing homes or congregate care facilities. And a large proportion of our tests over the past few months have come from colleges and universities. And in addition, as we look forward, we really are looking to address the K through 12 communities that we have. We also do a lot of business testing, including the Broad itself. So at the Broad, we test all employees that are on site, actually 3 times a week right now. And we've been able to do all of this at a pretty reasonable price point, in fact, far below the market average, charging only $25 per test for public and large-scale programs like our university program. We thought it was really important from the outset to be able to return results within 24 hours. So we'll talk a little bit about how we do that. And we've actually hired over 375 new folks at the Broad to help us get this testing off the ground and run 24 hours a day, 7 days a week. And so we'll talk through a little bit of these as we move through, but I first wanted to start with kind of the challenge and how we got here. And I can tell you, as Eric just mentioned, Niall and I both have spent many, many years at the Broad, really focused on DNA sequencing efforts, large scale, sort of groundbreaking resource building projects like the HapMap project and the Human Genome Project, and more recently things like TopMed, Human Cell Atlas. That's really sort of where our focus has been. And over time, we've built up a number of systems and infrastructure that has allowed us to do genomics at scale. But we found ourselves in March in a very unique position to have all of this infrastructure and systems in place, and to ask ourselves, what could we do and how could we pivot this entire group very quickly to address the needs that were happening at that time. And so to remind you, at that time, the US testing capability was pretty poor in comparison to other countries. On or around March 8th, this was a graph in news sources that basically outlined very low testing numbers for the US. And at the time, even the premier hospitals around us were maybe doing 400 tests per day. That was sort of the limit that they were at. And so we really set out to make a difference in a way that we could apply our learnings and our knowledge from our past decade or two of work on genome sequencing to this PCR viral testing. And so we decided officially to explore the test as a clinical diagnostic in our lab on March 9th. We basically looked at the CDC developed assay as sort of the starting place for our tests, but we repurposed as much of the existing infrastructure that we had in the lab as possible because we wanted to do things very quickly. So we sort of reallocated equipment, people, space to this testing effort, and we built out kind of the additional pieces. So we added walls to the lab in a matter of 24 to 48 hours to get the right biosafety requirements in place. We added a very light layer of software that would allow us to do clinical return of results as needed for these diagnostic tests. And then there was a number of activities that were required to obtain regulatory clearance. So of course we needed to CLIA validate, and we had previously invested quite a bit in building out a CLIA space and all of the quality system elements surrounding sort of what you would need to CLIA validate a test like this. We submitted to the FDA, and we'll talk a little bit more about that experience. And then we actually had to go work closely with the state to make sure that we could obtain the proper state licensing to handle the virology subspecialty that we were now going to embark on. And so we did all of that at lightning speed. So within the space of 2 weeks, we got all of the chemistry, all of the equipment, the people, all of those pieces together, and we were able to launch the test with a mere 1,000 tests a day by March 23rd. So this was a huge accomplishment. And once we started running those tests, we started to learn a lot very quickly about what it meant to run operations like this and what it meant to now interact with all different types of partners than what we had previously been accustomed to. And we'll talk a little bit about kind of some of those experiences next. But to give you a sense, the team itself, we repurposed a number of the scientists within our genomics group at the Broad and put them on 24/7 shifts, basically. So 12-hour shifts, 2 shifts a day, rotating 3 days on, 3 days off. Many of us spent time, in the middle of the night, many nights, making sure things were going okay. We also isolated the teams, so that way, in case one of the teams started to experience symptoms or we had a large number of positives on any particular team, we would have that contained. And we had backup teams assembled so that if one set of teams or one set of folks working on a team couldn't complete the job, we could ask others to step in. So that was a lot of very quick recruiting that we did. We did it within the group, and we also asked for volunteers across the Broad and had over 80 folks that work across the Broad join the genomics group for those first few months to help us get the teams up and running until we could hire more permanent staff. Over the course of time, we did hire 375 new people into the lab, and that number continues to grow over time as we continue to expand the lab. And here's examples of Niall, myself, as well as Stacy. We spent full days in the lab just filling tubes with media because that was the hardest thing we could get our hands on at the time, was just enough tubes to send out to the state sites so that they could collect effectively. So we kind of rolled up our sleeves and did whatever we had to do to kind of fill in the holes and the gaps as we saw them and as we observed them. So what I want to do now is hand it to Niall. He's going to talk a little bit about the assay and sort of what we're running in the lab today.
Thanks, Sheila. Yeah, so as Sheila alluded to, when we first started this, made the decision to start testing, we really didn't have a lot of time to go and explore all of the possible ways to do, SARS-CoV-2 nucleic acid detection. What we instead looked to is what was validated and available at the time. And so when we looked, the CDC had a molecular assay that uses this RT-PCR method that I'll discuss briefly. And so that is what we co-opted. And as Sheila said, we basically brought that assay in. We used a lot of the same reagents. We used exactly the same primers and probes, but we modified some of the kits and automation and ways that it was implemented in the lab, both to fit with the infrastructure we had in place, but also to help us get around some bottlenecks in supply chain. And supply chain has been one of the sort of maybe untold stories of the whole pandemic. From a lab testing perspective, there has been tremendous challenges across the board in securing reagents, equipment, and consumables to run this type of testing at the scale we're testing, because everybody in the world is trying to do the same kind of thing. So very briefly, at a molecular level, we can see, so on this slide, just a schematic of the SARS-CoV-2 virus, the pathogen responsible for COVID-19, and a image of its genome. It's an RNA genome, and at the very tail end of it, you can see a little gene called N, which encodes the nucleocapsid protein. And that is where the probes and primers for this RT-PCR assay were targeted from the CDC assay, and those are the primers and probes that we then used in our testing. So if you go to the next slide, Sheila. And so very briefly again, what RT-PCR is essentially a process by which we monitor the binding and amplification of the targeted primers to the genome of the virus or the human. So actually in each assay we run, we have two sets of primers and probes, one set that is targeted against the human genome, and that just allows us to ensure that for every sample or swab that we receive, that somebody has actually swabbed their nose, there is material there. So that acts as a sort of an internal control. And then there's also this set of primers and probes that target the N gene of the SARS-CoV-2 virus. And in RT-PCR reaction, we have these cycles of annealing of the primers. If their targeted genomic material is present, that primer binds and extends. Through the extension process, a bound probe, which sits between two of the amplification primers, is cleaved such that a fluorescence signal is released. And so we monitor in real time on the qPCR instruments the release of this fluorescence as we go through increasing cycles of amplification. And so therefore you'll hear the term CT. And so CT really refers to the cycle at which the signal from that particular probe becomes detectable above some threshold. And so that value is, even though in our assay we actually only report positive, negative, it is approved as a qualitative assay, not a quantitative assay. We can infer some things from looking at that number, that CT number. And what we've observed across all of the testing we've done is that actually while the human on the swab is somewhat consistent, the amount of virus we detect on a swab has a huge dynamic range. And this speaks to sort of the dynamic range of the assay itself. And so you can see from our distribution of positive samples in the red histogram here that we've seen samples with tens of billions of copies of the virus per mil of specimen, all the way down to individual copies of the virus per mil of specimen. So we actually detect sample across a huge range of viral loads within individuals. Show the next slide. So into sort of the application then. So we did the operational work, we built the infrastructure, we did the initial regulatory and compliance work to be able to do this type of testing. Then early in the pandemic in Massachusetts, one of the critical needs early on were the nursing home and assisted living facilities in the state. And it was recognized that these were places of extreme high risk because folks who live there and work there could be exposed, and also folks who were older were much more sensitive or likely to have bad outcomes from an infection. And so the governor of the Commonwealth did mandate early on that all staff and residents of nursing homes within the Commonwealth should be tested. And so we did partner with the Commonwealth and the National Guard. And so if you go to the next slide, you'll see the actual National Guard medic units were deployed on a mission to go and help collect swabs and specimens at all of the nursing homes and assisted living facilities in Massachusetts. So this was a huge mobilization of public resources to get these high-risk communities tested. When those swabs came back to us, we would have Humvees pull up at the front door and large coolers full of specimens pulled into the lab. We tested them. And one of the unique things about this first sort of nursing home testing program was everybody was being tested. Whereas in the rest of the country and in the hospital setting, the guidance at the time was that people who have symptoms should get tested, but testing wasn't indicated for people who didn't have symptoms. Whereas in the nursing home population, everybody was being tested, people who live there, people who work there. And so we actually had a unique view into what happens in folks who have symptoms when you test them versus people who don't have symptoms when you test them. And then on the next slide, we'll see what we actually observed in this cohort across tens of thousands of tests was that for folks who tested positive, there actually wasn't a significant difference in the amount of virus we detected in those populations. And this was important to understand at the time, and other folks had observed it in other settings, but it's important to note because it did guide us to, and public health policy to understand that there actually is importance in testing folks with no symptoms. And now, as we've gone through the pandemic, we know that many people who get infected with COVID will not have symptoms. But at the time, this was something of an insight that we were able to get from looking at the data we had generated. And so if we go forward, Sheila, back to you.
Yeah, so I'll jump in now and kind of, talk a little bit about, based on what we learned from that experience. We had a few months of running maybe 5,000 tests a day, in experience. And the nursing home experience was really critical for us because, we handled paper requisitions. So the information coming to us often would be handwritten on pieces of paper or handwritten on tubes. And so at one point, Eric asked us, would we consider and how can we think about really leaning in and really thinking about how we scale this lab? We had, a lot of discussions about how do we make this more, standardized, simpler? How could we handle thousands of pieces of paper coming in a day and then multiply that by 10? How is that going to be possible? And so we really had a moment in the spring where we had to step back and say, okay, let's think about even a full redesign of this process if we want to be able to scale this to a level, of 100,000 samples a day rather than just 5 or 10,000 samples a day. And so that brought us to, a new sort of phase in the way we thought about the test. And, to give you a sense of how it really differs from how other groups might think about the test, it's helpful to think, and kind of see how they're set up. And so, most of the large labs that do clinical diagnostic tests of this type often have a very non-standardized, non-uniform front end to their tests. And so often the materials are sourced by each collection site individually. They have their own sort of, choices that they make about the types of tubes or types of swabs that they use. In addition to that, often many labs, including hospital labs as well as diagnostic labs, choose highly specialized equipment to run these tests. So they might choose something like the Panther machine from Hologic that does a very good job of fully automating an all-in-one system, completing tests in a timely manner. However, it doesn't allow you to scale it substantially beyond what the limits of that box will do. In addition to that, and as Niall mentioned, supply chain was really at the top and forefront of our mind during this planning process. And groups like diagnostic labs or hospitals often, once they buy one of these sort of all-in-one systems, are sort of locked into the specialized kits that go with it. And what we were seeing from our hospital partners is they were basically at the mercy of these companies, getting kits to wherever, based on where the needs were greatest, that's where the kits were going. And so it was very hard for groups to kind of build up the supply chain they needed to address the surge or any sort of increase in positivity that was happening in their area. So this was basically our set of rules of what not to do as we built this new lab. We really tried to flip it on its head and think very differently about it. And so, one of the first things we did as we moved to a high scale, we started to think about what was the best way to actually collect these samples. And sort of the gold standard for many, many years was these nasopharyngeal swabs, the ones that sort of tickle your brain a little bit as they're administered. And we did enough studies and tested enough different types of swabs that we realized, we're not trying to culture the virus in this case, we're just trying to detect the presence of the virus. And often that can happen with just a very simple nasal swab, something that looks like a Q-tip and goes right in the front of your nose. Very easy, very simple, even simple for people to self-administer. And so this was one of the big sort of shifts that we had to make and work with the FDA on, was moving to these nasal swabs. In addition to that, as I mentioned, finding tubes with liquid in them at a scale high enough to be able to address the needs of the testing was becoming a huge challenge. And so we actually went ahead and validated dry swabs so that we could remove the need for liquid, and that greatly simplified the chain of custody and the details in terms of shipping these samples to our lab. And so now we wouldn't need to send out any media filled tubes. We could just send out kind of, basic tubes that were dry. People would put the swabs in them and return them to us. So that was a major fundamental shift for us as we moved to this new kind of improved scalable version. In addition to that, we sort of built out additional lab space. We took advantage of the space, the CLIA license space that we had at the Broad. And I'm gonna kind of put on in the background here a little lab tour as I talk through this. But basically you can see here the lab under construction. This is the same lab we used many, many years ago to sequence the first human genome. And today it's now where the majority of the COVID testing is happening at the Broad. And you can see here's what the tubes look like. But in order for us to really do this at scale, we knew we had to make it easy. And so one of the key design principles that we had in the beginning and still today is that we had to standardize the inputs and outputs of this test. So instead of taking paper requisitions from nursing homes, we had to work to find the right systems and build the right systems in place so that we could more quickly and easily automate this. And so you can see here, these are lots of different pieces of automation we have. This particular piece of automation, all it does is decap the tube, because at 100,000 tubes a day, manually decapping is actually a pretty lengthy process. So this machine decaps, about 1,200 tubes an hour, and we have multiple of them. But by standardizing the tube type, that made it very simple for us to now apply automation to this test in a new and sort of much higher scale way. And we've simplified that transport enough that it's dry swabs, that those tubes can come into our lab in a simpler way. They can come into very straightforward boxes instead of every sample being in a biohazard bag. We've removed all those biohazard bags, which seems very simple if you're handling 100 tests a day. But again, you don't want to be taking tubes out of 100,000 bio bags at a time. And so in addition to kind of those front end supplies and standardization of that input, we also focused on modular automation. So we took off the shelf, very well-known pieces of automation, things like the Agilent Bravo system or the Hamilton Starlet, pieces of equipment we had used for many years in our genomics work, and we repurposed them and bought additional to get the pieces of this process in place. And so you can see here, this is a step where we're doing the extraction. It's a very simple bead-based extraction that's been used many, many times over. And we're applying this in a 384 well format that allowed us to increase the density of the samples as they move through the lab. And we also tried to work with supply chains and suppliers that we knew well and we could take risks with in terms of making sure that they had very large volumes of the materials we needed. And so we worked closely with Thermo Fisher to supply many of their reagents for the tests. And we really aim to do that in a modular way. So if Thermo for some reason couldn't come through, although they have for us, we also validated additional master mixes or additional components from other suppliers. And we have at least 3 alternatives for every single step in the process that enables us to be able to manage any supply challenges that might happen as they come through. And so all of those modular pieces allowed us to really, kind of change the way, oops, sorry, let me get out of this. Change the way that we were able to, move through the testing at a very high scale. And so when we were able to get to a point where, we could hit that scale and turn that on, we had to sort of turn our, thinking to what kind of customer base would we now kind of expand this testing to, and how would we kind of think about this moving forward in terms of filling this line. And so, as I mentioned, that standardized input and modular process were really the key components. I'll just tell you one quick anecdote. even as you think about kind of the critical pieces of supply chain, things like these label writers, which are very simple. You can buy them at Staples or anywhere. We spent an entire weekend scouring all of Massachusetts Staples, looking for these printers as we started to bring on new customers and new sites so that we could get enough printers out to them. And so part of our
without her credit card that weekend.
Yes, exactly. So we just went and bought every label printer we could find, so we would enable testing sites that next week to be able to stand up and start running quickly. And you may ask sort of how we were managing across all of this, and we basically use a set of visual tools. And this is something I've worked at Sloan for a while with a number of folks. Sort of thinking about visual tools and clear targets as a way to make sure that we are on track and we can tell if we're ahead or behind based on the work that we're doing. And these are the types of boards we would have that we would sit in virtually, many of us virtual, trying to make sure everything was in place to hit our 100,000 tests a day. So I'm gonna skip forward to this, but basically what we were able to do was develop a set of steps that were highly automated and highly standardized that enabled us to reach that 100,000 sample a day target. And as we look forward, we now can continuously improve this process to see how we can expand it in the future. So I'll hand it back to Niall to talk a little bit how the FDA played into this.
Right. So at the start of this, as we've mentioned before, we already did have at Broad a clinical lab. And what that means, we were CLIA licensed by the state of Massachusetts and accredited by the College of American Pathologists. And we use that capability to do clinical research and translational work in genomics. We were able to leverage a lot of that same regulatory oversight for COVID testing. But at the beginning of the pandemic, the FDA also essentially claimed jurisdiction over this kind of diagnostic testing, but did provide a relatively lightweight mechanism for labs to be able to get started. And that's this emergency use authorization mechanism. This mechanism is in the news a lot recently because it is the same mechanism that Moderna and others are using to get approval for the vaccines. So there is an EUA route for testing as well. We were able in that March timeframe, when we talked about that 2-week period, once we had met our validation requirements for CLIA, for our clinical lab, we were able to start testing while we then in parallel worked on getting all of the paperwork in place and to the FDA. We just notified them that we were doing it. So that first mechanism is actually pretty lightweight and I think a credit that it helped a lot of labs get started sooner. As we start to evolve things and start to move out of sort of maybe the well-worn paths of what FDA had seen and the way things had been done in the past, moving to dry swabs, moving to nasal swabs, things like that, then it starts to get a little bit more complicated. And so, in the intervening months, we've had lots of back and forth with the FDA. We've generated a lot of data, clinical studies to basically convince them at each step of the way that what we were moving to was valid and safe and efficacious and met all the sort of sensitivity requirements that we needed for this test. So that's something we continue to do today. So as Sheila's mentioned, we have, and as we'll talk about at the end, we have some ideas to further increase scale and make testing available, even more available. And so we'll continue to work with the FDA to get that approved under the correct regulatory umbrellas. Okay, let's go move forward. So back to Sheila.
Yeah, so in order to achieve 24-hour turnaround time, we needed to think a little bit differently about how we run this process. So as you can imagine, we are an academic nonprofit research institute full of a number of scientists, but we really run this like a factory. And so we are using best-in-class manufacturing techniques. We are focused on the flow of samples through the lab, and we develop the capacity and design the work in a way that we are pretty sure, when every single sample comes in the lab, exactly when it's going to come off that line because of the design elements that we have in the lab. And so as you see many of the pictures in the lab or see the lab tour, you'll see that there's lots of colored tape on the floor in our lab. And these are really visual tools to trigger a response in the team in terms of what work needs to move forward and when. And so we use a very important design principle called pull in order to get the 24-hour turnaround times consistently that we have. And so, you know, the kind of classic sort of simple system and most intuitive system people have is what we would call a push system. So when samples show up, they sort of get pushed in, and you tend to have uneven inventory at various steps in a process. And, you know, that has a number of different, you know, sort of elements to it in terms of how the system will operate. One is that it often can be slow. And so what tends to happen is people start to call and say, hey, can you prioritize my sample and move it forward? But in a push system, you know, that would happen at very often at very many stages in the process, which would overall add time to each sample in the system. In a pull system, it's much more calculated, controlled inventory management system. And once a sample is into the lab, it actually shows up at our door, we don't allow any prioritization of work, which means it's first in, first out for every single sample in the lab. And that creates a highly predictable cadence. So there's no sort of special, you know, VIP samples in any way. They all go through when they arrive. That's how they move into the lab. And this allows us some very fast, high capacity processing. And so we've designed the lab around this pull system. We use those colored boxes as a way to kind of signal to the team whether more work needs to happen. So bring in and pull more samples in, or if there's something going wrong or there's an issue at one particular step, that colored box will get filled up and will signal to the team to slow down the earlier steps in the process. And that allows us to manage that process and reduce the overall turnaround time. So what does that look like? So for our proportion within 24 hours, we can hit 99% of the samples within that 24 hours. And the average turnaround times look really good. In fact, we've only had 1 or 2 instances in the past few months where we've had to either shut the lab down for a quality issue or found a particular problem that created a little bit of a blip in the turnaround time, but we were able to find those extremely quickly and address them within hours because of the system that we have in place. This is a particular challenge because most people collect roughly 9 to 5 during the day. So even though we're asking, the many colleges and universities to help us load balance, they still are going to send their samples roughly between 5 p.m. and 10 p.m. at night. And that's what we see in the data. So that means we're getting almost all the samples within a short period and then have to kind of work through that queue until the next day. And so we've adjusted the capacity in the lab to be able to manage that and still return all of those samples within 24 hours. And I'll turn it back to Niall to talk about the colleges.
Great, thanks, Sheila. So yeah, so all of this, and the investment in building up the capacity over the early summer months, a lot of that discussion was driven not just by discussions with the state and adjoining states on the needs in the community testing setting, but also Broad was approached by many of the universities and colleges in the New England area. And, with a simple question is, how can we bring students back safely for the fall semester? And so a lot of this, building capacity was in anticipation that we would be part of a frequent testing program at many colleges and universities. And so what we've seen is that we built this program, given the nature of, the need for sort of rapid turnaround and predictable turnaround time, it really was sort of I would say, quote unquote, local effort, really designed for New England institutions, although you'll see from the map that we did strain the definition of New England. We had New England and New England adjacent institutions, and through a lot of work across many groups, both just in the lab building capacity, but then with Sheila and Stacy and the legal team, the contracting teams at Broad, building outreach and having weekly seminars over the summer to really set up a system for ordering, for transportation of specimens to our central laboratory. And it involved things like working with courier companies to schedule routes to be really efficient, to pick up at the furthest universities and then pick up along the way and get back to our lab at a given time. And so that's what we had been running from the start of the fall semester. Many folks who may be watching might have been tested as a part of this program. You will be you are, I guess, you should be in some ways thankful that we were able to get the nasal swabs approved by the FDA so that your brains weren't being tickled twice a week. And so I would say in general, the program has been very, very successful. And so we had run, just under 4 million tests in the fall semester. Overall, the positivity rate in the universities and schools who were participating in this frequent testing program remained very low throughout most of that semester. And if you look at the data, which sort of shows schools who participated in frequent testing versus schools who did not, the outcomes were just so much better in universities and colleges who did participate in programs like this. And so as part of that, we successfully completed the fall semester. Most of the schools have many of their students now gone home for the remainder of this semester, but there is a plan to continue and pick up again to support the spring semester after the new year. And so if we go forward, one of the early things we learned is that we do need to continue to work and train institutions that we work with as to the appropriate means of collecting and submitting samples to the lab. It turns out that a red solo cup is not an approved method for submitting samples to your testing laboratory. We won't say which school this came from, but we don't exactly know what they were doing at their testing site. But many of the schools did really take their testing very, very seriously. And actually, one of the things that in the postmortem of this whole thing we'll see is the role of data analytics across the board. Within the Broad, we have an analytics team, and we can create our own internal dashboards to monitor many of our internal processes for quality control, for data review, for everything from billing and operations and supply chain to also, positivity rates within different testing cohorts. The schools, many of the schools also built similar programs. And, not surprisingly, in university settings, folks were really keen to sort of dig into their own data and built very elegant solutions to track outbreaks in their local institution on the campus, to do their contact tracing, to understand trends, and to monitor our performance in terms of turnaround time as well. So these are, people have really done great things in terms of just extracting knowledge from the data. And going forward, and again, just as we said, I think what we've seen now over and over in sort of testimonials from the universities who've been part of this and various stories that have come in the news, that it's, the testing itself does not prevent someone getting infected, but the act of testing doesn't do it. But the program of testing appears to actually impact people's behaviors and impact the way people think about their behaviors. And the fact that they are being tested so frequently gives them both confidence and security, but also reminds them that if they do something silly and get infected, they will be discovered relatively quickly. So that helps to modify behavior as well. And from the school perspective, the fact that you can test frequently and have results in a fast turnaround time allows you to plan to find those infections and very rapidly respond and isolate to prevent a small cluster from becoming a large outbreak. And that is a story we have seen repeated over and over on the campuses that we've been supporting throughout the last semester. And so, again, because these stories have come out now, we have seen this garner some interest. And so, the idea that, we should use this kind of testing as a model has gained interest all the way up to Dr. Birx, the White House coordinator, who visited our lab. Obviously in Massachusetts, Governor Baker also came to the lab. And so I think people have recognized that this kind of modality of testing is useful and could be broadly deployed and could be useful in other settings as well. So where to next? So given all of what we've talked about, and even with a vaccine on the horizon, which is great news for everybody, we do still see a need for increased testing, certainly today. We have a massive surge in the country. We're not immune to it in any part of the country. And so testing is more important now until as it ever has been. We really, even though there is a bright line on the horizon, we have to keep our eye on the ball today and not become lax in our behaviors. And something close to my heart as well is how do we make sure that people can be safe and confident in their schools, not just the universities, but in the K through 12 schools, and continuing to support nursing homes and other high-risk communities. And one way to do that is through surveillance testing. We'll talk a little bit about that. And so how do we actually support even more testing? It seems like with 100,000 a day, that should be more than enough, but actually we think we need more. We think we need more capacity. And rather than just building another floor on top of the building at 320 Charles Street, there's actually ways to do this with what we have. And so I talked at the beginning about the fact that today we actually, in our high throughput pipeline, look for two different targets. We look for a human to be present, and we look for the virus to be present. We do that in two separate reactions today. The molecular biology supports actually doing that in one reaction, where you can just apply a different fluorescent molecule to your human probe and your viral probe. And so that's called multiplexing. And so we could do that and immediately double our capacity on the detector end, on that qPCR end, from about 120,000 a day to over 240,000 a day with no additional equipment. So that's something that we're actively working on. If you were to do surveillance testing, and the idea, surveillance testing is slightly different from diagnostic testing, in that in surveillance testing, you're really looking for within a community, within a group of people, is there evidence of positivity? And so surveillance testing allows us to do things like pooling samples and test the pool of the samples. If the pool is negative, you can be fairly confident that everybody within that pool is negative. If a pool comes back positive, then you have an opportunity to tell a group of people to stay remote while you do individual follow-up testing to get to a diagnostic level for an individual. So a model, there's many ways to do pooling, but a model that we've been looking at would allow us to pool up to 10 swabs in a single tube. And again, that once that tube comes in the door, then it can be treated as a single sample through the rest of the process. So even taking a very small amount of our daily individual test capacity, we could do many hundreds of thousands of additional people in a surveillance mode. All right. And I think that was our last slide. And so just to sort of say thank you. And I think our next slide also just acknowledges the massive group of people across the board who've been involved in this. This has really been the work of a whole community to stand up what we've been doing. So I think we're good for questions and discussion.
Thank you, guys.
That's
I hope that was
fantastic.
fun.
Yeah. Yeah. So let me pose a few questions from the students for you. Of course, these are MIT students, so they want numbers. What are the typical false negative and false positive rates seen in your results?
I might jump in on framework for how to think about that question, and then Sheila and Niall can jump in. So false positive rates, how do you know? Well, one way you can tell is when we test colleges and universities, the actual positive rates were running well below 1 in 1,000. So that means the false positive rate can't be more than 1 in 1,000. Otherwise, the number would be higher than 1 in 1,000. Then when you actually check out those positives and see if they confirm, they really confirm. So you don't want to say you never, ever possibly get a false positive from the molecular biology, but the rate is very low, way below 10 to the -4. You could get false positives if there was cross-contamination, and some sites occasionally manage to do that, to cross-contaminate samples or things. So again, it's important to separate, is it a molecular biology false positive or some process false positive? Then false negatives, I want to make a firm point. It means it's a meaningless question unless you say, what's the false negative rate as a function of the true viral load? Like, if there's 10 to the 9th particles per mil, what's the chance you miss it? 10 to the 6th, 10 to the 5th, 10 to the 4th. So an average false negative rate is not a useful number. It's a conditional false negative rate, conditional on the true value in the tube. Although nobody talks about it this way. They always want to talk about what's your false negative rate, and it's some unholy integral over what happened to be the distributions of viral loads and things like that. And so, the question is, Niall showed you that distribution of viral loads that range over 9 orders of magnitude. And because we measure these sensitivities, you only get any meaningful rate of false negatives at the very end, the low end of the distribution. If there are only 100 particles per mil, the detection's not very good. 10, really not very good. 1 particle per mil, really terrible. But if you're above 10 to the 4th particles per mil, and certainly 10 to the 5th particles per mil, you have very good rates. So anyway, people never talk about a conditional false negative rate, but I feel like I should do that. Anyway, that's just my rant on how one should really think about these numbers.
Oh, and we needed that from a mathematician.
Yeah.
What were the trade-offs in the design of the test you run between sensitivity and specificity? How do we think about that?
Well, since the specificity is very, 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 very high, right? It doesn't produce many false positives. There's really not so much trade-off to speak of. So, Sheila and Niall should jump in, but it's not like, I mean, there is obviously in theory such a curve, but because RT-PCR run properly under the right settings produces few false positives, there's really not that much trade-off to think about in this setting.
Right. Another way to think of it is that you talk about the impact of those people who have very, very, very low amounts of virus, who you are more likely to tell them they're negative or miss that virus because it's well below the limit of detection of the assay. A single test is a snapshot in time. And so there's a big question in the community, of course, of what does it mean to have a certain amount of virus? And if you look at the clinical studies, people have shown that if you have below somewhere at 10 to the 6, 10 to the 5 copies of virus per mil, people have been unable to culture virus or have replicating virus below those levels. So then it becomes a question of that person may be infected, but are they infectious? And this is a big question that's happening right now. And then the correlate to that is also, this is also another reason why it's important to have a frequent testing program, because if you have someone who's very, very low, you've either caught them if you look at the time course of their infection, there's a very small chance you've caught them very early in the infection. There's a larger chance you've caught them in the longer tail end of their infection. And so that, even if we detect somebody who has a low amount of virus and you test them again 2 days later, their virus may be undetectable then. What you would want to do is, on the other end, capture someone who has a very low amount of virus and then 2 days later has a much higher amount of virus. That we see very rarely, but that is a reason to do frequent testing.
So really the thing to think about here is that when we talk about false positives and negatives, there's a technical question. Is this swab positive? Then there's a question, even if the swab is negative, might the patient be harboring virus that is not detected by the swab, or might they be shedding virus 2 days from now? So again, I think we think a lot about these words are easy to use, but you have to say precisely what you want to know about. And our biggest ignorance is about the public health implications, not about the molecular biological ability to detect the snapshot.
So I think there's a regulatory answer to this question that may make it moot, but it opens up issues for the future. In addition to extracting qPCR data on the virus from these nasal swabs, are there other types of data that are being collected or could be collected that might be informative?
In theory, you could sequence everybody, and you could construct the tree, and at a college, you could figure out who got an infection from whom. All those things are possible and could be really interesting. It has not been the focus of the testing lab, which has largely focused on serving, but there's a ton more data that in theory could be had that would teach us about provenance and transmission of virus.
Here's a disruptive question. Are there other more convenient testing methods that don't require sending samples to the Broad, i.e. point of care testing, being studied by you guys?
Well, I'll take it quickly because I know we're running out of time here. There's, for example, the Abbott BinaxNOW test, which is a point of care test, has to be administered by a professional, but you can get an answer in 15 minutes. And the question has been, how sensitive is it? It's less sensitive than PCR. But again, false negative rates as a function of viral load. So working with the state of Massachusetts Department of Public Health, we got thousands of samples from Lawrence General Hospital that were collected both with PCR for the Broad and Binax tests. And we have, paired data on them. So those data are available in an abstract that authors at DPH and Broad put out. And obviously there's a curve. It's less sensitive, but if somebody has very high viral load, it's quite fine. And so then it becomes a question of how do you deploy a less sensitive test that's at point of care versus a large scale test versus the pooled PCRs versus other things. And so we've only talked about one modality here, but really you have a bunch of levers.
Well, Sheila Dodge, Niall Lennon, and Eric Lander, thank you so much for doing this today. And please keep my tests coming back negative.
Well, you got to do your part, right? Our job is we're testing you enough that you think about it all the time, just like Niall said. But can I just give my thanks? I'm really here to cheerlead what Sheila and Niall and Stacy have been doing. They did this, and I believe all they asked for was permission. And I'm pleased to say they got it quickly, but they're true heroes for all of us in this New England area. And I'm just honored to be able to give them the opportunity to describe their work today.
Thank you, guys.